Hello guys, I'm Shubham of Tutorials. This is the part 5 of DBT JRF PYQs of 2024. Okay, so before we start, subscribe to our channel and press the bell icon to get notified whenever new videos out. This is absolutely free from your side, but it will help us grow. So first question, which one of the following antibiotic specifically inhibits RNA synthesis? Okay, RNA synthesis को inhibit करना है, मतलब transcription inhibitor, right? So basically, let us see the options. Isoniazid. Isoniazid is basically used for Mycobacterium tuberculosis, right? Treatment. So this is not for RNA synthesis. Penicillin antibiotic है, okay? Peptidoglycan synthesis को inhibit करता है. So this is also not the answer. As well as no isoniazid. Rifampicin, yes, ये आंसर है. Streptomycin is again related to translation. Okay, translation. So ये भी आंसर नहीं होगा. Option number 3, rifampicin is the answer. You can remember, I will give you a trick here. RNA synthesis, right? R for RNA and R for rifamycin. Remember this. Okay. Next question. Given below are two statements. Statement 1. If one parent carries the defective Huntington disease gene, his or her offspring have a 100% chance of inheriting the disease. Statement 2. Huntington disease is an autosomal dominant genetic disorder. So this is a question from genetics. How to solve this quickly? Basically statement 2 जो है that is correct. Okay, statement 2 is correct. I will mark here. Huntington disease है autosomal dominant. अबे autosomal dominant में क्या होगा? कैपिटल A स्मॉल a ये डिसीज होगा, एज वेल एज कैरियर भी, राइट? सो बेसिकली कैरियर इसमें नहीं होता, कैरियर इज इन द रिसेसिव केस, तो ये कंफर्म डिसीज होगा. ओके, व्हाट स्टेटमेंट वन इज सेइंग? इफ वन पेरेंट कैरीज द डिफेक्टिव हंटिंगटन डिसीज जीन, दिस इज अ वन पेरेंट कैरिंग अ डिसीज जीन, ओके, his or her offspring have 100% chance of inheriting the disease. How 100%? When gametes are formed, either capital A will be inherited or small a will be inherited. अगर capital A inherit होगा तो disease होगी, right? So this is half chance ना, and ये तो normal है, तो 50-50%, right? Not 100%. Okay, 100% कब होगा? When the disease is autosomal recessive. Okay, autosomal recessive में disease के लिए both the alleles are required for the disease, right, to manifest. Okay, and ये carrier होगा in autosomal recessive, right? But in autosomal dominant carrier नहीं होगा, disease ही होगा. Disease and carrier are same. Okay, so statement 1 is wrong, right? Statement 1 is incorrect and statement 2 is correct. Option 4 is the answer. Okay, next, which one of the following diseases is due to severe deficiency of proteins in diet? Okay, protein diet deficiency. Kwashiorkor. Yes, right, Kwashiorkor. Tay-Sachs disease. This is a lysosomal disorder, okay, where the hexosaminidase A gene is mutated. Okay, so this is not a protein. scurvy is vitamin C disorder, and myasthenia gravis is an autoimmune disease, राइट. सो क्वेश्चन जो ऑप्शन 1 है इसका आंसर होगा. नेक्स्ट, which one of the following is responsible for development of myelin sheath in the central nervous system? Okay, astrocytes, no. Astrocytes are basically involved in blood brain barrier. Okay, 3 B's you should remember: blood brain barrier, astrocytes. Oligodendrocytes, yes. Okay, oligodendrocytes form the myelin sheath in CNS, right? CNS myelin sheath. And जो Schwann cells हैं that form myelin sheath in PNS. Okay. Microglia, these are for the phagocytosis, right? Phagocytosis. Scavenger cells, we can say. And dendritic cells, dendritic cell again a role in immunity, right? But not in a development of myelin sheath, right? So ऐसे हम solve कर सकते हैं questions. How to solve quickly, basically that I am showing you. Next question, which one of the following disorders lead to hallucinations? Okay, hallucinations. Anxiety, no. Schizophrenia, yes. Alzheimer's, no. Epilepsy, no. Okay, so option 2 is the answer. Next question, in absolute refractive period of neurons, Na plus, sodium plus channels are open, sodium plus channels are closed, potassium channels are open, potassium channels are closed. Okay, now this is from action potential, very important. Okay, I will just give you some images. You can make a screenshot. Majority of questions can be answered from that. Okay, now see, ये slide action potential. See, first absolute है, refractive period, and then relative. Okay, then what is in absolute period? You can see here depolarization हो रहा है, right? Depolarization. Na plus channels are open and K plus channels are closed, right? This is absolute period. Got it? Na plus channels open. So sodium comes inside cell and positive involvement होता है, right? So the answer will be option 1. Na plus channels are open. Next question, phagocytosis in the central nervous system involves. Okay, phagocytosis में क्या होगा? Recovering excess calcium plus from synapse? No. Specialized ability of some neurons to divide? Phagocytosis में division होगा? No. Bridging the blood-brain barrier? No. Clearing of dead cells? Yes. Right, जो dead cells है उनको clear करना, और इसमें रोल है माइक्रोग्लिया. We saw. Okay, related question है, right? Quantitative trait loci can be mapped using transgenic approach, SSR markers, gene editing, tissue culture. Tissue culture नहीं होगा. Quantitative trait. Okay. and this is basically related to loci. Loci means what? Specific location on a chromosome. ओके मतलब ये लोकेशन है, तो इससे हमें आइडेंटिफाई करना है। तो टिश्यू कल्चर से नहीं होगा, जीन एडिटिंग से नहीं होगा, ट्रांसजेनिक अप्रोच से नहीं होगा। एंड ऑप्शन 2 SSR मार्कर्स, दिस आर वेरी इंपॉर्टेंट फॉर क्वांटिटेटिव ट्रेट लोकाई, मार्कर्स हैं। ओके, कोडोमिनेंट मार्कर्स, SSR मार्कर्स आर कोडोमिनेंट। गॉट इट? तो ऑप्शन 2 आंसर होगा इसका। नेक्स्ट, टारगेटेड जीन डिसरप्शन कैन नॉट बी अचीव्ड बाय। जीन डिसरप्शन मतलब जीन को इनएक्टिव करना है। ओके, नॉकआउट। जीन डिसरप्शन मीन्स वी कैन से नॉकआउट। क्री-लॉक सिस्टम, यस पॉसिबल, हमें कैन नॉट देखना है, राइट, कैन नॉट। क्रिस्पर सिस्टम, यस, जीन एडिटिंग करके हम जीन को डिसरप्ट कर सकते हैं। जिंक फिंगर न्यूक्लियस, यस, इससे भी पॉसिबल है। ZFN। cDNA इंटीग्रेशन, नो। टारगेटेड जीन डिसरप्शन पॉसिबल नहीं है cDNA इंटीग्रेशन से, इट इज नॉट दैट स्पेसिफिक, राइट। तो स्पेसिफिसिटी में क्रीलॉक्स, क्रिस्पर, ZFNs एंड टैलेंस भी है। Okay, remember this. Talents also come in gene disruption, right? So answer will be option 4, tDNA integration. Next question, which is the last one. Centimorgan is defined as the genetic distance between two loci with a statistically corrected recombination frequency of. See, genetic distance में centimorgan is related to recombination frequency, that is percentage of recombination. Okay, so 1 centimorgan is basically 1%. 10 centimorgan is equal to 10%. Right, 5 centimorgan है मतलब 5%. So this is equal. Okay, suppose it is 50 centimorgan means 50%. तो इसका आंसर होगा 1%. Okay, question is asking centimorgan is defined as, means 1 centimorgan is equal to what recombination frequency? So it is 1%. Option 3 answer होगा. Okay, so this is how you can solve questions. Basically you can analyze the options, check the options, and option elimination से भी आप answers कर सकते हो in the quickly manner, right? So that's it for this video. I hope this video helps you. If you find this video helpful, do let us know in the comment section. Like this video, share this video with your friends, subscribe to the channel, and stay tuned for the upcoming videos. Thank you.